Hello everyone. Today we will discuss about isoenzymes or isozymes. Isoenzymes are different forms of the same enzyme. These are different forms of same enzyme. These are having different amino acid sequence or different genes. So isoenzymes are different forms of the same enzyme having different amino acid sequence or different genes. But they catalyze the same reaction. Isoenzymes differ from each other either structurally, electrophoretically or immunologically. So let's see some examples of isoenzymes. First one is lactate dehydrogenase, LDH. LDH catalyzes the reversible oxidation of lactate to pyruvate. Lactate to pyruvate. Five isoenzymes are there. LDH 1, 2, 3, 4 and 5. All these isoenzymes catalyze the same reaction, that is the conversion of lactate to pyruvate. These different forms can be separated by electrophoresis. For example, this LDH1 is the fastest moving fraction in electrophoresis. It is the fastest moving fraction. Then LDH5 is the slowest moving fraction in electrophoresis. Then these isoenzymes have different physical properties. For example, LDH4 and 5, these are easily destroyed by heat. Then LDH1 and 2, these are heat resistant. Then these isoenzymes have different Km values. They have different Km values. So mainly 5 isoenzymes are there. Then a sixth atypical isoenzyme is found in male genital tissues, that is known as LDHX. So this is an atypical isoenzyme of LDH that is present in male genital tissues. Then each isoenzyme is a tetramer. And it consists of 4 polypeptide subunits, which consists of H and M units. 4 polypeptide subunits and it consists of H and M units. So 5 possible combinations are there. That is LDH1 is H4, LDH2 is H3M, LDH3 is H2M2, LDH4 is HM3 and LDH5 is M4. Here LDH1 is the fastest moving fraction in electrophoresis. It is the fastest moving fraction. It is mainly found in myocardium. Then LDH2, that is H3M, it is found in reticuloendothelial system. LDH3, it is H2M2, it is present in lungs. LDH4 HM3, that is in kidneys, then placenta and in pancreas. Then LDH5, this is the slowest moving fraction in electrophoresis. It is found mainly in muscle, liver and intestine. So in normal condition, LDH2 is the prominent enzyme. It is the prominent enzyme, LDH2. Then after, so LDH1 is the specific marker for. So after myocardial infarction, LDH1 is the specific marker for myocardial infarction. So after myocardial infarction, LDH1 increases. So LDH2 is the most prominent isoenzyme in normal condition. After myocardial infarction, LDH1 increases. Myocardial infarction, this LDH1 increases. So LDH1 is the specific marker of this myocardial infarction. Then in the case of acute viral hepatitis, this LDH4 and 5 increases in acute viral hepatitis. Then total serum LDH, it is increased in neoplastic diseases. Then in malignancies, there is usually an increase in LDH3 and LDH3, 4 and 5. In malignancies, these 3 increases, 3, 4 and 5 increases. So that is about LDH. Mainly 5 isoenzymes are there. LDH1 is the fastest moving in electrophoresis, LDH5 is the slowest moving in electrophoresis. Normally, most prominent isoenzyme is LDH2, but after myocardial infarction, LDH1 increases. So LDH1 is the specific marker for myocardial infarction. So that is about LDH, lactate dehydrogenase. Next one is CK or CPK. CK or CPK, that is creatine kinase or creatine phosphokinase. This creatine kinase catalyzes the conversion of creatine to phosphocreatine, creatine to phosphocreatine. It's a reversible reaction. Mainly 3 isoenzymes are there for CK. 3 isoenzymes. And it is a dimer. It is a dimer. It is composed of B and M units. B for brain and M for muscle. So the combinations are CK1. It is BB. Polypeptide chain is BB. It is the fast moving and it is seen in brain. Then CK2, it is MB, it is specific to heart. Then CK3, it is MM, it is the slow moving and it is mainly present in skeletal muscle. So these are the isoenzymes CK1, CK2 and CK3. 3 isoenzymes CK1, CK2 and CK3. It is a dimer, consists of B and M units. B for brain, M for muscle. CK1, the combinations are CK1, it is BB, polypeptide chains. These are the polypeptide chains. BB, that is the fastest moving. CK1 is the fastest moving fraction in electrophoresis. It is mainly found in brain. CK2 is MB, it is found in heart. Then CK3 is MM, it is a slow moving fraction and it is found in skeletal muscles. In myocardial infarction, CK2, there is an increase in CK2 within 4 hours. Increase CK2 within 4 hours, maximum in 24 hours and then falls rapidly. In myocardial infarction, there is an increase in CK2 occurs within 4 hours. Then maximum in 24 hours, and it will fall rapidly. This CK2, it is about 4.5 to 20% of total CK activity. It is about 4.5 to 20%. It is in plasma of this recent myocardial infarction. And the total CK2 level is elevated up to 20-fold above normal in myocardial infarction. So, mainly 3 isoenzymes are there, CK1, CK2, CK3. Then atypical isoenzymes are there. These are known as macro CK and mitochondrial CK. Mitochondrial CK. These are the atypical isoenzymes of CK. So, that is about CK, that is creatine kinase. It catalyzes the conversion of creatine to phosphocreatine. It is a reversible reaction. Mainly 3 isoenzymes are there. In myocardial infarction, CK2, there is an increase in CK2. So, next one is ALP, that is alkaline phosphatase. ALP, alkaline phosphatase. Mainly 6 isoenzymes are there. Alpha 1, alpha 2, alpha 1 is seen in biliary. Then alpha 2 is heat labile. Heat labile, it is found in liver. Alpha 2 heat stable, it is in placenta. Then pre-beta, it is in bone. Gamma in intestine, and 6th one is leukocyte ALP. These are the 6 isoenzymes of ALP. This ALP is used for differential diagnosis of jaundice. It is used for differential diagnosis of jaundice. Then very high values are seen in xanthomatous biliary cirrhosis. Xanthomatous biliary cirrhosis, very high values are seen. Then in the case of hemolytic jaundice, serum ALP level will be normal in hemolytic jaundice. Hemolytic jaundice, this level will be normal. It is mainly present in this ALP is mainly present on biliary epithelium. It is on biliary epithelium. So, these are the 6 main isoenzymes. Then atypical isoenzymes are there. These are Regan isoenzyme and Nagao isoenzyme. Regan isoenzyme and Nagao isoenzyme. These are known as carcinoplacental ALP isoenzyme. Carcinoplacental ALP isoenzyme, because they resemble this placental isoenzyme. Because they are, that's why they are known as carcinoplacental ALP isoenzymes. This Regan isoenzyme is produced by malignant tissues. Then they are found in carcinoma of lungs, colon and ovary. Then Nagao enzyme, Nagao isoenzyme, it is detected in metastatic carcinoma of pleural surfaces. Metastatic carcinoma of pleural surfaces, and in adenocarcinoma of pancreas and bile duct. Adenocarcinoma of pancreas and bile duct. So, these are the atypical isoenzymes, Regan and Nagao isoenzymes. These are known as carcinoplacental ALP isoenzyme, because they resemble the placental isoenzyme of ALP. So, that is about ALP. The next one is hexokinase. Hexokinase, mainly 4 isoenzymes are there. 4 isoenzymes, that is hexokinase 1, 2, 3 and 4. In this hexokinase 4 is known as glucokinase. It is known as glucokinase. So these are the common examples of isoenzymes. Then based on their origin and formation, four forms are there. That is true isozyme. First one is true isozymes. Second one is hybrid isozyme. Then third one is allozymes, and the fourth one is isoforms. So first we will see, so based on origin and formation, four forms: true isozymes, hybrid isozyme, then allozymes and isoforms. So first we will see true isozymes. True isozymes are the products of different genes. These are the products of different genes, and these are coded by different genomes These are the products of different genes and they are coded by different genomes. For example, malate dehydrogenase enzyme. This enzyme has two isoenzymes. Cytosolic and mitochondrial. That is true isozymes. Next one is hybrid isozymes. These hybrid isozymes are oligomeric proteins. These are oligomeric proteins produced by different combination of the constituent subunits. Different combination of constituent subunits. That is known as hybrid isozymes. For example, LDH. LDH has 5 isoenzymes. LDH 1, 2, 3, 4 and 5. That is hybrid isozymes. Then third form is allozymes. Allozymes are variant of an enzyme. These are coded by different alleles of the same gene. So allozymes are variant of an enzyme. These are coded by different alleles of same gene. For example, glucose 6-phosphate dehydrogenase. About 400 isoenzymes are there for this glucose 6-phosphate dehydrogenase. The next form is isoforms. Then this isozyme and allozyme. These isozymes are usually results from gene duplication. But in the case of allozymes, these result from point mutation or by insertion deletion. That is isozymes and allozymes. The next one is isoforms. Isoforms are a result of post-translational modification of the same gene. These are as a result of post-translational modification of same gene. For example, ALP. ALP has 6 isoenzymes. That is isoforms. So next we will see. Based on origin and formation, 4 forms are there: true isoenzymes, then hybrid isoenzymes, allozymes and isoforms. Next we will see properties or characteristics of these isozymes. 。 So, the difference between some isoenzymes are due to difference in quaternary structure of the enzyme. Difference in quaternary structure. Then these isoenzymes have different kinetic parameters. For example, Km values. Then electrophoretic mobility. Electrophoretic mobility. For example, isoenzymes of LDH have different mobility. For example, LDH1 is the fastest moving and LDH5 is the slowest moving fraction. Then next property is heat stability. Heat stability. For example, isoenzymes of ALP. These are either heat labile or heat stable. Isoenzymes of ALP. Then next property is antibodies. For example, CK. For CK, each isoenzyme can be bound only by a specific antibody. Each isoenzyme can be bound only by a specific antibody. So, these are the properties or characteristics of isoenzymes. So, this is about isoenzymes or isozymes. This is today's topic. Thank you for watching.